Today, we'll be looking at engineering bugs and the wild world of synthetic biology for conservation. If you're new to this channel, welcome. This is Mr. Singularity, where we explore the scientific and technological breakthroughs shaping the future as we know it. Imagine a world where a mosquito bite is merely an irritating itch. No malaria, no dengue fever at all. Scientists revealed last month that they had taken another step in that vision, explaining how synthetically modified mosquitoes avoid the spread of dengue fever, a contagious tropical disease that affects as many as 100 million people per year. Now, imagine genetically tweaking an invasive species of mosquito to save native Hawaiian birds from extinction, or moving genes from one vulnerable species of chestnut tree to another to help the latter avoid a blister. Using the same kind of genetic modification that is used to make a plant-based burger bleed, scientists are starting to investigate how synthetic biology can help preserve habitats and restore animals. Synthetic biology, or Synbio, uses the new and most effective gene editing techniques, such as the cut-and-paste technique known as CRISPR-Cas9. In tandem with modern techniques for digitizing and automating the design and simulation of different genetic components, science can now manipulate species to generate novel food ingredients or to rewire switches that transmit genes that regulate those functions. In the case of the dengue-carrying mosquitoes, scientists genetically engineered members of the Aedes aegypti population by passing genes from the human immune system that produced an antibody to combat dengue fever to the blood-sucking bug. The antibody is triggered and expressed as soon as the female mosquito draws blood. In effect, the mosquito is cured by dengue fever before the disease can be spread. The next step will be to propagate a new genetic factor to confer dengue immunity to the population. This is where the gene drive comes in. Gene drive mechanisms, which can be natural or synthetically engineered, have warped the inheritance of a certain genetic variable so that it spreads more rapidly over decades. The aim is to circumvent standard inheritance rules, the classic Darwinian hypothesis that inheritance is driven by genetic differences that enhance the ability of the individual to compete in the dog-eat-dog world, so that re-engineered traits become dominant. In terms of conservation, Synbio could theoretically solve a range of areas of interest, such as constraining invasive species, reducing the impacts of wildlife trafficking, improving disease tolerance, and even taking the species back from the verge of extinction. Biologists at the University of California, who also headed the team that published the paper on mosquitoes, have developed a novel gene drive method to control genetic inheritance in Drosophila suzuki, a fruit fly widely known as spotted-winged Drosophila. This specific insect, native to Japan and first found in the US in 2008, injects its eggs into soft ripening fruits such as berries. Existing practices to protect against spotted Drosophila depend on either heavy insecticide use or early harvesting. The pest is expected to cost the US economy as much as $700 million in damages per year. The engineered gene drive, named Medea after the character in Greek mythology that destroyed her offspring, uses a synthetic toxin and accompanying anecdote feature to achieve 100% inheritance bias in less than 20 generations. This genetic Trojan horse could then be used to disperse elements that impart susceptibility to certain environmental factors, such as the death of modified fruit flies at a certain temperature. UC San Diego Associate Professor Omar Akbari told Singularity Hub that his team is close to field tests on some of our technologies. The most far-reaching solution for our community will be the use of precision-directed sterile insect technique to monitor the wild populations of D. suzuki. A variety of businesses are turning to Synbio to manufacture foods where the natural commodity is costly, uncommon, or endangered. Take the well-recognized example of vanilla. Many products on the market use a synthetic version of the main ingredient vanilla manufactured from petrochemicals. Swiss group Evolva has produced a genetically engineered yeast to manufacture vanillin in a close manner to that of brewing beer. Modern Meadow also uses DNA editing techniques to design advanced collagen-producing yeast cells for the manufacture of leather products. In a case more specifically related to wildlife protection, Singaporean scientists have designed a synthetic substitute for horseshoe crab blood cells, which have been used in biomedical applications for decades. Both 4 species of horseshoe crab are considered to be vulnerable by the International Union for the Conservation of Biodiversity. However, though a substitute medication for horseshoe crab blood has been commercially available for more than 15 years, It is yet to be universally accepted for a number of purposes. This is slowly evolving as recent experiments have shown that available synthetics are just as reliable as horseshoe crab blood for the identification of endotoxins in biomedical development. The long-lived American chestnut tree was once one of the dominant tree species in the eastern U.S. The blight from Asia, launched at the end of the 1800s, has all but washed them out. Efforts to breed American chestnuts with disease-resistant chestnut trees in China have had little results, as it is not easy to disperse the desired traits from many genes over the next generation. A project led by the College of Environmental Sciences and Forestry in Syracuse, New York, is using Synbio to grow blister-resistant American chestnuts without damaging the fungus. Researchers also copied a single gene from wheat and passed it to the American chestnuts. The gene produces an enzyme called oxalate oxidase that does not kill the fungus. Instead, it breaks down the fungus toxin that destroys the tissue properties of the tree. The bonus is that the fungus itself is left untreated, meaning the blight stays dormant and does not develop resistance over time. Although bringing the dead back to life is one trick that is likely to elude science in our lifetime, Synbio experts have been vigorously trying to revive the woolly mammoth and other endangered animals, such as the passenger pigeon, which vanished more than a century ago. These ventures are not simply making pure specimens of these long-lived animals. Rather, scientists are introducing parts of ancient DNA code into modern-day relatives. In the case of mammoth fur, researchers are seeking to build a mammoth-elephant hybrid using an Asian elephant. Proponents of this kind of resurrection research say it's not about attempting to restore dead animals, than about preserving others who are actually at risk of vanishing. The Asian elephant, Elephas maximus, is on the IUCN Red List of endangered species. A team led by George Church from Harvard University hopes that by bypassing genes in the mammoth genome in the Asian elephant, it'll be able to live in the Arctic. The genes involved could include those that code for extra fat and thick fur. This will expand the range of species to areas that are now shifting due to the warming of the climate. Like geoengineering, manipulating the atmosphere to present some of the consequences of climate change, bioengineering has its critics and critics. Some respond viscerally in some way to the concept of altering natural processes. One of the key arguments is the fear that the creation of a genetically engineered species may have adverse effects. While no one expects the Jurassic Park scenario, where genetically enhanced creatures are pursuing Jeff Goldblum through the jungle, there is a possibility that genetically modified traits could leap species or somehow go off the plot. Kent Redford thinks it is important to promote a dialogue about the potential advantages and drawbacks of the conservation position of the Synbio, regardless of where one stands on the divide. The mission is to ensure that the conservation community is aware of and informed about these technologies and try to influence these technologies for the sake of biodiversity, to minimize harm and increase positive outcomes. Redford is the chairperson of the IUCN Task Force on Synthetic Biology and Protection of Biodiversity, a conservation specialist who has worked at the Nature Conservancy and Wildlife Conservation Society. He was editor-in-chief of the evaluation paper Genetic Frontiers for Conservation, which will be discussed this summer at the IUCN World Conservation Congress in France. IUCN's viewpoint counts. Its 1,300 affiliate groups include states, non-governmental agencies, industry societies, and research and scholarly institutions. Redford refused to comment about what kind of suggestions could come out of the IUCN conference. He acknowledged that the junction between Synbio and conservation lies on the fringe for those in the conservation world. Most of my colleagues don't see why they should pay a lot of attention to this. Any of us who are aware of these new developments consider them to be relevant tools to help solve some of the essential conservation problems. Others claim that these genetic methods have the potential of completely destroying the natural world and the lives of poor people. The key obstacle for conservation Synbio is not technology, but the obtaining of regulatory approvals in the public interest. I think we need some time. As more technologies are being developed and tested with positive outcomes, we believe the resistance will decrease. Although the scientific community is discussing the promise and pitfalls of symbiotics, biodiversity will begin to decrease. Last year, a report from the United Nations Intergovernmental Science Policy Forum on Biodiversity and Ecosystem Services published a series of troubling figures. For example, the total abundance of native species in most of the major land-based ecosystems has declined by at least 20%, mostly after 1900. And almost 10% of all domestic breeds of human mammals used for food and livestock in history have been extinct by 2016, with at least 1,000 more breeds remaining at risk. What's your take on this? Do you think the natural world is in serious trouble? Let me know down in the comments below, and check out one of these other videos. This has been Mr. Singularity, and I'll see you on the next one.